So before I'm going to start these presentations, everything is covered by these two eminent scientists in marine science. That carotenoids is an importance, is a natural pigment, and also many fungus and bacteria produce it. So what is the my take and why I'm presenting these bioprospecting angles? Because by expert, I'm a plant biotechnologist. I did my PhD from Calcutta University, and that time I'm working on golden rice project. So golden rice is a project where we are putting carotenoid biosynthesis pathway cloning from the bacteria to rice by specifically endosperm specific promoters. So my expertise is on editing of genome, and also tissue culture brings me formulation of different media. So I'm not preferring to use any common media. I formulated myself because it's a challenge on bioprospecting, because you can't bioprospecting on available commercial media because it's not feasible. So let's start something, and definitely I'm not putting so many data because yet it need to be verified by multiple you can say the experiments. So what I have, it gives you clear idea, and also the youngster with the encouragement that what I am found is little bit missing in many. So let's go. So as I said, I'm a plant, so I choose first seaweed because it's very close to plant, although it's not a plant, it's a microalgae. So I chose isolating associated fungi, which are maybe epiphytes or endophytes. Epiphytes those are the outer surface, mostly pathogens type of thing, or maybe in flow they come and stuck. And endophytes, those are within that. And one thumb rule is that endophytes are good. They have association for a longer time, and they can help each other. So as I'm also working on host pathogen interactions between the rice, jute, and other hosts, so it is also my interest. So luckily we got a project from DOM, from MOES, and it's a good funding project. So I start something looking on that. So what is the important here? You can see that sometimes few fungi are not so common. It appear, and sometimes it appear on by chance. Sometimes it's choose the media condition. Sometimes it choose the temperature condition. So it's very tough to isolate endophytes. Okay? So I not only endophytes, I also isolate many fungi from different sea level. Okay, these are the epiphytes from sediment sample. So different samples, and I isolate mainly fungi. So my media is formulated in such a way that it only allows the fungi to grow, and it has the antibiotic, so there is no chance of growing of the bacteria. So next, sometimes it's very difficult to isolate the endophytes because sometimes it's submerged with the others fungi. Like here, you can see that the red dots are, sometimes we are not able to find it because it's just submerged with some others fungi. So it requires multiple streaking and multiple time you need to do culture to get it. So next part is, there is a good PNS paper on that how we to understand. So it's a thumb rule to check the ITS. It is the locations where you can see that the part of ITS, and here just by PCR and clone that part of fragment to plastic and NCBI, you can get the idea about genus at least, or genus and species sometime. And sometimes I also found out some similarity check. It has to be 98% and above, but sometimes I got only 40%, only 30%. Either I am the first isolating it, or maybe no one submitted the ITS sequencing NCBI. As I mentioned, this is our Calcutta University's work, where these are my mentors there, and I clone most of these genes for the rice biofortification. That's why now I'm coming to my actual interest in marine. So these are the two parameters I'd like to interest, that the lipid market is growing. Okay. And this lipid market, India has a 6.4% growth rate, and carotenoid market also growing. So these are the main two components. And why lipid and carotenoid? Because carotenoid dissolved in lipid. So when you wanted to bioaccumulation of carotenoid, you need some lipid. So this is a perfect combination, and most of the fungi are producing it side by side, but not sometimes produce a lipid higher amount. Some cases they produce carotenoid higher amount. So my interest was there to find out some microorganism which produce simultaneously. Okay. So this is because my talk is the initial talk, and after that there are a lot of talks on human health and cancer and all. So I fit it everywhere. You can see these are all papers on the carotenoid. You can see these are the specifically fungal carotenoids paper. So it has everything like antioxidant, antiviral, anti-inflammatory, antifungal, antibiotic, diabetic. And luckily I'm from the ILS, and ILS has all the facility to check whether and how it is worked. So let's start, and today I'm trying to focus on these two fungi, and these are the yeast. One is the Rhodotubula and one is Pichia. This color shows that it is the lipid producing, and as I mentioned it already. So it is proved that these are producing lipid, and profile of lipid is different. So this is the GCMS data showing that the composition of their lipid. So now is the carotenoid. So and every carotenoid producing fungi is not same. So you can check, we have a 4 type of carotenoid isolated from seagrass, some seaweed, sediment, and they are producing different amount of beta carotenoid and lipid also. So we are isolating this lipid, go for the spectra, and we found that they are producing not only beta carotenoid, also others important lipid. So you can check that in the UV light, you can take the others lipid also. So it produces beta carotenoid. Obviously, we know the function of beta carotenoid. And here is the interesting part is that food is the limiting factor nowadays. India is the highest populated country in the world. So we should not competition with the food for that type of minerals. So carotenoids from the other source will be the help in the nature. So Rhodotubula produce similar amount of carotenoid like carrots. So it's my dream. Can we produce carotenoid for human consumption and not from the food source? So can we produce it from the marine sources? So yes, lot of things need to be done. But yes, these are the benefits. And this is the recent paper with producing that claim. So I'm looking for it. Now, few months, we are testing carotenoids from different perspectives on different feed trials and all to check it whether we can go in directions to produce carotenoid from marine source and for human consumption. So I want to thank WSC and also DBT and the MOES for giving us the fund. And I'm open for questioning and open for collaborations. So I'm here. After the talk, we can also talk and take a few questions. Thank you, sir.
Thank you, Dr. Lajunta.